Okay, I'm going to record this
Okay.
because I don't know that Maddie, if she has a migraine, is even going to be able to participate, because I think I don't get migraines, but I understand that they can be very, very nasty, and that even hearing things can set them off. So
Yeah.
I'd be surprised if she signs in. So let's get one thing out of the way instantly, and that is your counseling for next mod. You have two classes. You're going to have Pharmacy 209 with Nate,
Okay.
and you're going to have COM 103, which is Computerized Pharmacy Systems and Databases, with Shelley Christie, who's a new instructor.
Okay.
Shelley and I have talked. She's our new billing and coding instructor, so she's used to working with these kinds of databases. I've given her some hints on dealing with the software.
Okay.
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 So that's what that is about. And let's see. I'm just going to go ahead and assume that Maddie's going to be able to hear this. Maddie, you're going to have phlebotomy next mod, and that's going to be Monday and Wednesday evenings. Nate is trying to contact you. Nate Crowder, your instructor, please get in touch with him. He's got 5 classes to teach. He's trying to schedule. So
Give me one second.
Okay, I'll wait till you're back, Brandy.
Oh, you can continue with Maddie.
Okay. We need to have you talk to Nate about the schedule. There's only 2 of you in the class, and we need to get you scheduled for a time that you can actually do the phlebotomy class. So please do contact Nate, and it's Nate nathan.crowder@collegeamerica.edu. So I think he's tried to contact you already, but go ahead and contact him. So Brandy, I'm going to close the door to this room so I'm not lecturing the hallway.
Okay. That's fine. I need you to be reading.
Okay. So I know you've had kind of a rough time, but you've got some work to catch up on. Are you going to be able to do that?
Yeah, I'm gonna work on it tonight. Hopefully get a lot of it knocked out. And
okay. Yeah,
it's just been hell.
Yeah, well, we're very very sorry to see this going on. So we will be covering a couple of other things today. The lecture tonight will be pretty short because, mostly it was going to be you guys doing catch up anyway.
Okay.
I was thinking we'd try a catheter insertion, but I have a funny story on myself.
Okay.
To tell. Last week we gave a lecture. I gave you a lecture about catheters.
Mm-hmm.
On Saturday, I needed to be catheterized.
Oh no.
Yeah. Talk about ironic.
Yeah.
They even got a laugh in the emergency room, which is good. You don't get a lot of laughs in an emergency room.
Not really, no.
But yeah, they all thought that was funny that I had just done this and I had to be catheterized. So It's been an interesting weekend.
I can
Not as
see.
interesting or stressful, I suspect, as for you, but nonetheless, it
Yeah.
was interesting. I finally got to learn what I talk about.
Well, that's good. Unfortunate circumstances where you had to learn, but
Yeah, well, it's really weird when that happens, but I sort of figure it's justice.
yeah.
So let's do a quick look at what's going on this week in this class. Um, you're supposed to read chapters 6, 9, and 45 in the textbook.
Okay.
For what
That's the medical assisting book?
Yep, the big
Okay.
black boat anchor.
Okay.
Okay. The discussion is about CLIA testing. CLIA is the Clinical Laboratory Improvement Amendments passed by Congress in 1988. And I discussed that in my post, so I won't repeat all that stuff since you're going to read it anyway. But you're discussing the 3 levels of CLIA testing.
Okay.
That are there. And so you'll have to do a little research, but what we do in our labs is the waived section, which is the easiest, the least complicated of the assays.
Okay.
And there's more data that you'll see on in what I posted on that. So now if we go to the assignment, we're looking at the biological hazard and safety guidelines from OSHA, which you guys remember from Med 102
Mm-hmm.
and your OSHA bloodborne pathogens training.
Mm-hmm.
You may want to brush up on that. And then the whole issue of total quality management requirements, or TQM management requirements in the laboratory. So this will require some reading, and some of the stuff in the black textbook covers that as well.
Okay.
But just writing a short, basically 300 words is about a page. So
Okay.
just write that brief paper on that. TQM, OSHA regulations, as you remember, are a requirement, and OSHA can do some nasty things to employers who break the rules. One of the nastiest to me is something that does affect a medical office, which is, what do you do with sharps containers? OSHA can assess a fine of $7,000 per container if you fill beyond the fill line.
Jeez.
Yeah, that's not a trivial amount for a medical office. It may not mean anything to a hospital, but actually it does. But it is a significant thing to consider. So, these OSHA guidelines are there, and you've all been through it, and you've been following them. I mean, gloving up and, the whole PPE stuff. In reality, if you were working in a lab and testing things like urine, you might be required to wear a mask and goggles just for protection, because we don't know whether urine theoretically is sterile, but we don't know that about anything.
Yeah.
So we have to make the assumption using universal precautions that anything can be contaminated, potentially contaminated biological materials.
There's one resident that we have, well, actually we have a couple that have VRE.
Oh,
Yeah.
that's no fun.
Yeah. So for like the longest time, it was like you need your PPE, you needed a gown and glove every time you did the catheter drain or toileted them.
Yeah.
And then it switched from gown and glove to just gloves. And we're like, you all have like instilled in our brains like how like contagious this is. And then you're just like, oh, well, it's not that bad.
Yeah.
So like everybody panicked after that.
Yeah, I bet. Yeah, that doesn't make any sense. And in fact, You know, an OSHA inspector might have some words to say about that, or a state inspector.
Yeah.
So, you know, that's something to remember. You know, I don't understand decisions like that. This is why I think decision making is so poorly carried out in so
Mhm.
many organizations.
Yeah, because I mean, they were like, "Oh, well, you know, it's not that bad. We don't need the gowns. We just need the gloves." And like probably 3 days later, a catheter bag for the resident that has it leaked all over the pants of one of our employees.
Right.
And she had to go and like go to the doctor and get examined and like sign this waiver and like all kinds of stuff. And we're like, if it's not that bad, like why why are we doing this?
Yeah. No, that just demonstrated that it is that bad.
Exactly.
So yeah, that's not too cool. I find it very difficult to understand why decisions like that are made.
Hmm.
Because it's not that expensive to have those things, to have the
It's
gowning,
not.
and it's a lot cheaper than ending up with a patient or an employee who's been harmed.
Hmm.
So, oh, by the way, I have a note here on your schedule that you need to have that we need to have proof of your TB status.
Okay.
TB check. You should have had one recently in the last year working where you are.
I didn't actually.
You may want to get one before you start your externship.
Because I remember that I got one when my son was like 2.
Well, he's a bit
No,
older than that now.
no, he wasn't even 1 yet. My ex-husband
Huh?
was just deployed. And he ended
Okay.
up getting TB on his way to Guam.
Oh,
And so I had to get tested.
yeah.
And that was the last time I've had a TB test.
I'm surprised that they don't have you get one working at the home.
Yeah, I don't know.
Well, it's going to be a requirement before you start that, so you
Okay.
may want to get it scheduled when you can.
Yeah.
Okay. All right, so let's move on to the next topic. We have obviously the week 4 assessment, which is in effect, you know, a summary. You've got this little quiz called Career Integration. Please do that. And our in-class activity this week was going to be this, to create a review of the class that for you to post, send to me so I can post so the class can see it. You can use your whatever format you want, and then submit it as a document to me. And then once I have all those, I will make them available for you to access in an announcement or some other format that I choose, and will let you know. Why
Okay.
is that? Because what you will do at next by the end of the week is actually review one of your obviously Maddie's, you know, and Maddie will do yours, but you'll complete a review of that. And then it's really due by the beginning of class time on Wednesday. But go ahead and do that.
Okay.
All right. So, and I'm going to trust that Maddie gets all this so that it will be done. Then you have this quality management thing, which is another Medline University course,
Okay.
right? That you'll be doing. And then finally, our own final.
Okay.
For this class. The final should be set up. I'll make sure it's set up so that you can do it at least a couple of times.
Okay.
All right. So let's go into the lecture because I'd like to go ahead and get that done. Since you obviously have work to do. So quality assurance and quality control are two important concepts. What are the quality assurance part is really looking at the quality of their products. Do they meet the specifications? Do they actually perform a function in a predictable and trustworthy manner? Okay, so quality assurance is a lot of different things. It can look at what you do to assure that that you meet the standards that are required. Now, what this means in a setting of a laboratory associated with a medical office or medical testing is that the quality assurance part is what happens when you is that if you follow those procedures, the company is telling you that they have tested this batch of product and that it meets the specifications that have been approved by the FDA. All of these tests are approved by the FDA. We've been doing all the tests we've done for everything, the flu, the strep, the urine for pregnancy drugs, etc. All of these are approved by the FDA as being appropriate for the assessment of the health of a patient and the determination of any sort of treatment that they require or a disease state or some other or the need for further testing. So that's part of that. The company that manufactures it is telling you that it has met their quality standards. In the pharmacy, I think you may remember we talked about the U.S. Pharmacopoeia as being a standards for manufacturing of pharmaceutical products. And again, if something says that it meets USP standards, that's a quality measure. You are assured that the product is what it says, has the potency it says, and is safe to use within the time frame of its dating. Okay. In the case of a test, you're assured that the results are going to have an almost immeasurably small probability of being in error. And that is especially the ones that you'd be using in an office, like the ones we've been doing here. We've been using these CLIA-waived tests because they are high quality and reliability, and we can trust them. But all of them have, have some way should have some way of testing them. And in fact, with one of the things that we didn't pull out of the drug kits because they're expensive things, is that we have a way of actually spiking the sample to show whether or not the tests reveal the presence of drugs. That's a QA standard. And it's something that if you get a batch of materials, you're often encouraged to do a quick QA test to see if the tests are working properly. And what they do is they have a positive and a negative control, meaning that you'd do two tests, one with the positive control, one with the negative. And what do we got here? Right. No problem, Brandy. No problem. You know that. So you should do those just as a means of confirming that the batch is good. And if it fails, then you have to return it right away. rather than use it, obviously. And by failing means that you get the wrong results from the controls. Okay. And the other part of quality assurance is these are the procedures that are in place within an organization for the manufacturing or preparation of a product. Manufacturing,
quality assurance
processing, packaging, and the policies are established to make sure that throughout the lifetime of the product is going to function exactly as measured. This holds whether you're talking about a drug or a test, or for that matter, a Honda. Every manufacturer of anything will have a quality assurance group that will be certain that the product rolling off their line is going to work the way they think and the way they want. As a program, it also, for in the medical field, is a program that guarantees quality in terms of patient care, and then and watching and taking reports on how the product is working in clinical practice. Sometimes products go out there and you've done all the research you want. It looks like solid gold, and you get out there and you get millions of uses and you suddenly find out there's a problem. This is a bigger problem with drugs than most of our CLIA-waived tests, because the tests themselves have very little room for variation. Whereas a drug, the variation comes in patients. And so, and in the development of a drug, for instance, we're always looking at safety, and it's a statistical game that we play when we're looking at the safety of a drug. And that statistical game is looking at the incidence of side effects or adverse reactions, and making some judgments that the FDA has to agree with about whether or not those side effects or adverse events are frequent enough to make the drug too dangerous to use. The same with tests. We expect that any of these CLIA-waived tests, we have a 99.99% probability of the test being error-free, because tests are used for diagnostic purposes and for making plans for treatment. So we want those to be accurate. So we look at those. So what we're looking at is something that that the quality of our service or product meets certain standards, and it meets them consistently. If we have a manufacturing process or any process that gives us variable results in terms of the output, then there's a problem there. In fact, in the medical business, we have what we call good manufacturing standards, good manufacturing procedures that we follow. And those are established separately for every product, because every product is different. So that's part of setting up our QA system, so that we have a consistent process. So so whenever we order a test, we have to be careful about paying attention to that detail. We talked about this as a process earlier in the class. We've got to look at the tests being ordered and why, and the sample being collected properly, stored properly, transferred properly to the testing lab. Most places where you give a urine sample at your doctor's office, it's going to be tested there, and it's probably going to be tested virtually immediately. When I had to go in to the ER to get catheterized, I think that when they finally got a urine sample out of me, I had the results in 5 minutes, which you can understand having done those tests now yourself. There's no reason for it not to. And fortunately, the ER was pretty unbusy. I think I was one of two at MCR in Loveland. I was one of two people in the ER.
That makes sense.
Yeah. So, everything happened in a hurry. But we look at the, and this, they're talking here about phlebotomy, but it's the same thing holds for urinalysis. We want to know what happens in the lab where the test is done. The lab needs to be a clean environment. It doesn't have to be sterile, usually. A clean environment, and the actual test has to be run within the proper time frame, under the proper conditions, and absolutely according to the manufacturer's instructions. Those instructions are there for a good, solid reason. So all of that, and then what happens, the documentation and what happens with the information, who gets the information after the analysis is critical as well, because there's a reason it's been ordered. So we've got these. So we look at an ordering process, collection, transportation, handling, and the completeness of patient information. It's critical to be sure we have that. Sometimes automated systems are used. We don't obviously automate what we do here, but there is a Clinitec device that you can put your urine sample in and you get a readout. And so it's an automated analytical process that then gives you a printed result.
That's pretty cool.
And that's probably what happened to me this weekend, because everything came back on a printed page, which was fine. And of course, confirmed exactly the same thing we measured in class. So that was nice. So the analytical activities include the equipment. If it's an instrumented process like the Clinitec, then that stuff needs to be regularly tested and QA'd. You need to be testing it on a regular basis to be certain of the accuracy of the readout. The test strips, the waved stuff, are actually, as I say, we can QA them before we start using them, but they're pretty reliable. But the reagents need to be fresh. The supplies need to be available. You have to have people who know how to use the equipment, and then they need to do the job correctly. No sloppiness, none of that. And then the general stuff in the lab. What happens when people go in QA and analytical lab, they'll look around, and if they even see water bottles in the lab, they'll get unhappy.
If state comes in and we have water bottles.
Sorry?
If state comes in and we have water bottles on our med carts, it's fined.
Yeah.
Like, we cannot have water bottles on our med carts.
No, you shouldn't. And it's the same in a lab setting. We allow you to bring water into the lab. Our lab is different. A teaching lab is a little different. But for the most part, you want to be careful not to have any consumables in the lab. That's why we have break rooms and water fountains. And any other activity that's going on, sloppiness, the lab is dirty. Labs need to be clean. So, and the thing about automating things is as long as you maintain the equipment, the smaller the chance of error. And errors, most of the errors that occur using these kinds of tests are human error. They are either sampling, storage, transport, or the way the sample is fed into the instrument. Many times these things are being designed now to alert you when things aren't right, if there's not enough sample. We see this a lot with the finger stick test. If you don't get enough blood onto the little card, it won't give you a reading, which is good. It needs a certain amount. And afterward, we need to see the reporting and the data. It needs to go into the patient chart. It's just amazing. This morning I called my primary physician, and they just pulled up my chart and told me what went on. Of course, I knew what went on, but it was nice that they had all that. So we want to be sure that everything goes to the right physicians or right practice. And any referrals go to the right place as well, along with those reports. And it needs to be timely. This used to be a problem. You might wait for a long time. I mean, I remember in the old days when you went down to get a CT, it would sometimes take days to get a reading. Now it's overnight, if not sooner. And part of the reason it's overnight, to be honest, is that guess who does the reading? You go down for your CT in the afternoon, and that information is then shuttled electronically to some place like India, where you have a trained, a UK or US or French or German trained radiologist who reads it, and then the report is back the next morning.
What?
Yeah. Yeah, all of this stuff it can be done locally, but this is how a lot of them are doing it now because it's cheaper. Bangalore is a heck of a lot cheaper than Denver.
True.
Yeah. So, and that's exactly what happens. And the reason I know this is that years ago, our daughter had a head injury, and we were getting the MRI and CT scans back overnight. And I went to the neurosurgeon who was monitoring the case, and I said, "Jerry, why is this going on? I mean, how can this be so fast? This is thorough." And he says, "Oh, it's red in Bangalore." And he said, "In fact, I know the radiologist doing the reading." So, it's
What?
done. Yeah.
I never knew that.
Oh yeah, it happens. It's done a lot. So but now things are so fast, they go straight in. I had to have an MRI done a year ago, or earlier this year rather. And it was done at UC Health. And by the time I got home, it was already in my UC Health, My Health folder. So it's that fast. So again, we get these things, and timely is critical. Some things, if your urinalysis is an hour late, it's probably not going to be fatal. If your blood work is real late, it might cause problems. Okay. So also we want reference ranges in all these reports so that everyone, especially the patient, because patient sees a number. Number is meaningless. A range at least lets them understand that it can vary, and the number has a little more meaning, even if they haven't a clue what the numbers themselves mean. I see this in my family a lot. So we have to have a way of measuring the effectiveness of our QA system. And usually that is looked at averages or percents. And what that means is the average error rate, and either as a percent or as a or as number per 100,000 tests or something like that. And so they look at it. So it gives it, and using these numbers usually tell us ways that we can look at improving that quality, finding out where the problem is. When there's a failure, that's the question most researchers are going to ask. What happened? Why did it fail? And then that investigation then helps reduce the potential for that kind of failure again. That's pretty standard actually in engineering for that matter. You know, why did it fail? Okay, how do we fix it? All right. And then we look for the next failure. Okay. Threshold values, we have that with the dipstick test for urine. There is a threshold value below which we cannot read a result. And so that's sort of the minimum that we have. And then we have what we consider to be the upper range of that threshold value, that above which or below which there could be a patient problem. That's what the reference ranges are. They're thresholds. Are they fixed? More or less, because they too are averages. Remember, we were looking at, what was it, bilirubin, 0.6 to 1.2 for men.
I think so.
And 0.5 to 1.1 for women. So there
Yeah.
are differentials, right? Remember that?
Mm-hmm.
So anywhere in that range is considered fine. Where we start looking is when we start getting to the extremes of that range. And then we start sort of watching. So those threshold values are something that we don't want to see. What we also are looking for in part of our testing of the original product and part of our QA testing on each batch is that we will deliberately expose the product to samples that are outside the threshold and make sure that that shows. And if there's a problem, then we have to fix our manufacturing. Okay. In terms of patient care, the example they're giving here, declining patient care in an ICU, it may be that we're inadequately staffed, or it may be that there are some other problems in the ICU, some environmental problems perhaps. Number of years ago, let's see, 1978, 79, there was an outbreak at an American Legion convention in Philadelphia of a mysterious bacterial disease that caused fatal pneumonias. And it turned out this was the Legionnaire's disease.
Oh, crap.
Caused by an organism that we now call Legionella that was in the water handling system of the hotel. It took ages to figure that out because they didn't have the tools, and they couldn't overcome the biases they had to figure out that there was a reason that guests at the hotel were getting sick, but workers weren't. Because the workers had already been exposed and were immune. So they didn't get sick. The guests, mostly elderly, most American Legion members are elderly. The elderly are much more prone to pneumonia and to pneumonic disease that kills them. So it was really, it took a while to figure that out because everyone figured it was a closed system. Well, guess what? Okay,
That's great.
documentation is a large part. When the state or anyone else comes into your facility, one of the things they look at is your documentation, and they really go after you on that.
I almost had a
I've
med
already
error.
seen that. Pardon?
I almost got written up for a med error. I had to go back to work. I left work and then had to go back to work 3 minutes later because I almost forgot to sign out a narcotic.
Ah,
Yep.
yep, yeah, you know that. And in fact, one of the things that sort of makes me angry is that many states employ contractors to do that investigation, and they pay them by the ding.
Mhm.
And I think that is just BS.
Mhm.
They shouldn't be paid by the number of infractions they find, because then they find a lot, and some
Mhm.
of them aren't infractions. So it's kind of interesting. But we want to be sure that all of these things are handled at a very high level of proficiency, because they may help you in court. Years ago, my wife was working in her first job as a PT in a hospital in Raleigh, North Carolina. And she was there on a weekend doing a weekend rotation when in came a patient who was brought in. He had been racing a boat on a lake a few miles east of Raleigh. The boat flipped, and he experienced a very high level break, spinal break, and he was a quad. And Susan treated him and gave him all the PT that was requested, and worked with him, helped him, and then she left that job. And 5 years later, just the week before the statute of limitations was going to run out, we got notification from his lawyers that they were suing the hospital and every single person in the hospital that had anything to do with him. It was one of those blanket lawsuits that they filed at the deadline because they didn't do the timely work of figuring out what was going on. They also sued the boat manufacturer, the race organizers, the EMTs who dragged him out of the boat and saved his life, the helicopter pilot who airlifted him to the hospital, everybody. So this, of course, made my wife very nervous. And I had been through some of these, not in medical stuff, but in contract negotiations that I had been a part of on several occasions. And I said, don't worry about it. Just go in and answer the questions. Well, again, her documentation saved her. Her documentation showed that she had done everything that was A, ordered by the physician, and everything that was B, considered standard care for someone with this kind of injury, and they dropped her from the case, which they should have.
Yeah.
But it was that careful documentation that saved her. So those needs to be known. Now, quality control is basically doing what that quality assurance plan laid out. The quality assurance plan says this is what we do. The quality control part is that we actually go through and be certain that everything is done exactly correctly and documented, and all the tests. I don't know if you remember a number of years ago, this would have been, I was back, would have been in the 1980s, or you may have heard about it. In Seattle, a woman took some Tylenol and died. very shortly thereafter. And when they did the autopsy, they found that she had died of cyanide poisoning. And so they went in and they were testing things in the house, and they found cyanide in a bottle of Tylenol. So Johnson & Johnson immediately pulled cyanide. All the pharmacies in Seattle is where this occurred, started pulling Tylenol off the shelf. And Johnson & Johnson did some testing, and some independent labs did some testing. And there were a number of bottles of Tylenol in several manufacturing batches that were contaminated with cyanide.
How the hell did that happen?
Well, the story goes, and it continues, gets interesting from here. Johnson & Johnson, what they did was they got the batch numbers. They did a nationwide recall of those batches, and they started testing. The only place that there was any cyanide found in anything was Seattle. None of the others from that batch that were in other areas of the country showed any trace of cyanide. They also went back to their QC records, their quality control records, and looked at the documentation, and the products were documented and shown to be manufactured according to the good manufacturing procedures that were required, and according to the approved manufacturing method that they had for Tylenol. They also could not find any evidence of cyanide in any of the stored samples from those batches that they keep by law and test quarterly to make sure that the product maintains its stability and quality. What happened was this woman's husband decided to kill her, and contaminated cyanide into the Tylenol. And in order to try and cover his tracks, he went around and bought up a bunch of Tylenol from a bunch of different pharmacies, contaminated them all, and then surreptitiously put them back on the shelves in those pharmacies. Somebody remembered that this guy had come in and bought a bunch of Tylenol, and that's how they connected it to him. And I sincerely hope they put him in the chair. But nonetheless, I thought, why didn't you just divorce her? You'd still be not in jail. But that's why those records are critical, and why the testing is critical. So as we say, part of our quality control is that we calibrate our machines and we follow their calibration procedures. When we're working, when I was working in as a research chemist, we had a lot of equipment. Some of it you'd think it's fairly ordinary, balances and things like that. But we would calibrate those things on a regular basis to make sure that we got accurate weights, so that when I reported in my lab record that I weighed out 1.35 grams of some substance, I could be assured that it was 1.35 plus or minus 0.00001 gram. So we really do that, and we calibrate them the same exact way, because different calibration techniques can give you different results. So it's just like people who take their blood pressure at home. They should always take their blood pressure at exactly the same time of day. Otherwise, their activities are going to affect that blood pressure. I used to tell my mother-in-law, get out of bed, go to the bathroom, and then go take your blood pressure, and record it. And she did, largely because she used one of those automated things that I think suck in general. But
yeah.
But it but you know, the lab has to have, if your lab has to have a way to control the accuracy of the procedures for analysis. So we're looking at here again. The example here is phlebotomy, but it's the same for virtually anything. We're looking at patient prep, identification, the equipment, to make sure that you know, we use the proper tubes in the proper order, and that we make sure that we let each tube be completely filled per its vacuum ability. No tube will fill fully, but we want to be sure that we get the vacuum exhausted. They need to be labeled so that we know, and you know how they do that at 9Health. They've got little stickers with numbers. The numbers are keyed to a barcode, which is keyed to the patient, so that we can really keep track of that. Technique is critical, making sure that you use that, and then looking at the priorities. And this is going to change with some patients. Some patients will have they'll need stat draws for whatever reason. Usually they're looking at some reaction to a potent drug that they're trying to monitor. We cannot, the terms QA and QC, unfortunately, are conflated sometimes. We can't do that. Basically, I like this analogy, lawmakers and police. It doesn't do any good for the lawmakers or the QA to assure the public we live in a safe neighborhood if there aren't any police to enforce the laws, right? Those are the QC side. So that's the thing about, excuse me, that's the difference here. But QA and QC are often exchanged, and what it amounts to is that QA is the promise and QC is the delivery. Okay, the promise is this is what we're going to do, and then the delivery is that this is what we've done and it matches our promise. Okay, does that make sense to you?
I actually was a quality control manager for a point of sale system company.
Oh.
So yeah, I listened to all the recorded calls, and yeah.
Well, you should have given this lecture.
I should have, but it's fine. You're the instructor.
Well, then do you have anything to add to that? Did I miss anything important?
No.
Okay. Good. Let's just, it's one thing, you know, when we're talking about medical technologies, the need for QA and QC is probably an order of magnitude higher than many other things. Now, not every other thing. An airplane, for instance, is possibly even higher.
Oh yeah.
But you know, that sort of thing it means that we're doing things that can influence the health and well-being of patients, of human beings. So what we want to do is make sure that that our products are of the highest quality, and that we have procedures in place to make sure that every step in the process of delivering whatever it is, it's first off, it's delivering a test to you, and then you delivering the results to the people who need to know them, the patient and the caregivers or the care providers. Those have to be, we have to be 99.99% confident in those. And that takes work. And this is where some places fall down. Scrimping on doing the QC part of all of this. The manufacturers are going to do that, but then once it's on site, you're responsible party for storing the reagents or the reagent strips, for cleaning and maintaining the equipment. I mean, even an autoclave, we have to clean periodically. And you'd think that an autoclave, by definition, should be sterile, but it isn't. There can be all sorts of things in there. So even those should have a regular cleaning cycle on them. Any place that you store samples should have a regular cleaning cycle so that you can be sure that that sample is reliably not going to be contaminated by other substances. And the handling, everything. It's really kind of it's something that is a hidden part that people don't understand, is a hidden part of making certain that we deliver properly. We try to train, all of us in this business, try to train you to understand that these things have to be done properly and have to be done the same way at all times so that we can rely on the results. We have no room for people who are kind of loosey-goosey about these things. And in fact, if they're there, they need to go do something else.
True.
That's my opinion. Anyway, any other comments or questions?
Mm-mm.
Okay. You can call this up anytime, by the way, since it's in the shell. So I'm going to back up here, and we're approaching an hour. So I'll get this converted and posted. Let's see, you still have a few things that you need to do from last week.
Yeah.
So go ahead and get those out of the way.
Okay.
We're going to cut this off in a couple of minutes. And so Maddie, send me an email when you read this, and I'd like you to include the word Borneo, B-O-R-N-E-O, in the email to show that you've listened to this. Same with you, Ariel, if you happen to listen to this. Also, I suggest you go ahead and do your daily checkpoint tonight.
Thanks for the reminder.
Because I know it's easy to forget those. Let's see. So the quality management Medline University course, do the in-class activity and get that posted to me. And then when you get to your assessment and the career integration, you should be, this will be the writing one. You've done this before. So this will be the writing one. And you should also have done your questionnaire for the mod questionnaire already for this week. So any questions or thoughts?
No.
I'm sorry that you can't be in tonight. And I don't think we're going to get a chance to play with our little friend lying in the bed in MedLab 2.
That's okay.
Put a catheter in her because I don't think we'll have time on Wednesday.
That's okay.
But we'll see.
Okay.
Okay?
Mm-hmm.
So if there's nothing else, we'll just go ahead and end this for tonight.
Okay.
All righty?
All right. Thanks, Burke.
You're welcome, and I hope you can get things worked out. Go ahead and contact Amanda.
Okay.
And see if there's anything she can do to help.
Okay.
Okay?
All right. Thanks.
So take care and good luck.
Thank you. I'll see you on Wednesday.
Okay, great. Look forward to it.
All
And
right.
see you on Wednesday too, Maddie, I hope. Okay, bye.